After studying this module, you will be able to know about the significance of drug elimination, the concept of elimination of drugs from the body, and the different ways of drug elimination. The capability to proficiently remove toxic materials is critical to the subsistence of a species. The intricacy in toxicant elimination processes has increased proportionate with the increased complication associated with animal form. For unicellular organisms, passive diffusion can be sufficient for the elimination of toxic metabolic wastes produced by the organism. Similarly, as exogenous toxic materials derived from the environment diffuse into a unicellular organism, they can also readily diffuse out of the organism. The disposition of a chemical or xenobiotic is defined as the composite actions of its absorption, distribution, biotransformation, and elimination. The disposition of any compound is an essential factor that contributes to its potential for toxicity. Specifically, the toxicity of a substance is directly dependent on the dose, where dose is defined as the amount that ultimately reaches the site or sites of action such as tissue, cell, or molecular target. Therefore, the disposition of a chemical determines its concentration at the site of action such that the intensive actions of absorption, distribution, and elimination also determine the potential for antagonistic actions to occur. Toxicants are eliminated from the body by several routes. The kidney is perhaps the most important organ for the excretion of xenobiotics because more chemicals are eliminated from the body by this route than by any other route. The second important route of elimination of many xenobiotics is through feces, and the third, largely for gases, is through the lungs. Biliary excretion of xenobiotics and their metabolites is most often the major source of fecal excretion, but a number of other sources can be significant for some compounds. All body secretions appear to have the ability to excrete chemicals as toxicants, and drugs have been found even in sweat, saliva, tears, and milk. Next is drug disposition theory. Toxicokinetics includes the more appropriate study of exogenous compounds, that is xenobiotics, the adverse effects related with their concentrations, their passages through the physiological compartments, and their ultimate fates. Compartmental toxicokinetics involves administration or exposure to a chemical, rapid equilibration to the central compartments, followed by distribution to peripheral compartments. Consequently, the principles of pharmacokinetics and toxicokinetics historically have been used interchangeably. First is one compartment model. A one compartment model is the simplest illustration of a system that refers to the first order extravascular absorption of a xenobiotic. The absorption rate is constant and permits for entry into central compartments, that is plasma and tissues. Few compounds follow one compartment modeling. Agents such as creatinine equilibrate rapidly, distribute uniformly throughout blood, plasma, and tissues, and are eliminated quickly. Drug or toxin elimination processes generally follow zero order or first order kinetics. First order elimination and first order kinetics terms describe the rate of elimination of a drug in proportion to the drug concentration. An increase in the concentration of the chemical results in an increase in the rate of elimination. This typically occurs at low drug concentrations. On the other hand, zero order elimination refers to the removal of a fixed amount of drug independent of the concentration. Increasing plasma concentration of the chemical does not proceed with a corresponding increase in elimination. Zero order kinetics usually occurs when biotransforming enzymes are saturated. Clinically, this situation is the most common cause of drug accumulation and toxicity. Next model is two-compartment model. In a two-compartment model, a xenobiotic chemical requires a longer time for the concentration to equilibrate with tissues and plasma. Essentially, molecules enter the central compartment at a constant rate and may be eliminated through a constant first order elimination process. However, a parallel reaction occurs when the chemical enters and exits peripheral compartments through first order rate constants for distribution. The distribution phases are inconstant but rapid relative to elimination. Most drugs and xenobiotics undergo two or multi-compartment modeling behavior. Next is urinary elimination. The kidney is a very efficient organ for the elimination of toxicants from the body. The functional unit of the kidney is the nephron, containing the glomerulus, a capillary tuft that initiates the process of filtration of the blood, and tubular elements within the renal cortex and medulla that function to produce and concentrate the urine. Toxic compounds are excreted in urine by the same mechanisms the kidney uses to remove the end products of transitional metabolism from the body, including glomerular filtration, tubular excretion by passive diffusion, and active tubular secretion. In general, the excretion of small molecular weight water-soluble compounds is favored in urine. Depending on the physiochemical properties of a compound, it may be reabsorbed across the tubular cells of the nephron back into the bloodstream. Thus, toxicants with a high lipid or water partition coefficient are reabsorbed efficiently, whereas polar compounds and ions are excreted in urine. The pH of urine may fluctuate, but it is usually slightly acidic. Toxic agents can also be excreted from plasma into urine by passive diffusion through the tubule. This process is probably of slight importance because filtration is much faster than excretion by passive diffusion through the tubules, providing an advantageous concentration gradient for reabsorption rather than excretion. Exceptions to this generalization may be some organic acids and bases that would be largely ionized and thus trapped at the pH of urine. For renal excretion of such compounds, the flow of urine is likely to be essential for the preservation of a concentration gradient favoring excretion. Thus, diuretics can hasten the elimination of weak organic acids and bases because they increase urine flow. Xenobiotics can also be excreted into urine by active excretion. This process involves the uptake of toxicants from the blood into the cells of the renal proximal tubule with subsequent efflux from the cell into the tubular fluid from which urine is formed. Transporters may be expressed on the apical cell membrane where efflux pumps contribute to tubular secretion and influx pumps are important for reabsorption. Transporters localized to the basolateral membrane serve to transport xenobiotics to and from the systemic circulation or the renal tubular cells and also contribute to reabsorptive and excretory processes. As many functions of the kidney are incompletely developed at birth, some xenobiotics are eliminated more slowly in newborns than in adults and therefore may be more toxic to infants. For example, the clearance of penicillin by premature infants is only about 20% of that observed in older children. In contrast, some compounds such as cephaloridine are nephrotoxic in adult animals but not in infants. The reduced toxicity results directly from the lack of active uptake of cephaloridine by the kidneys in infants, such that the chemical is not concentrated in the tubules. Urinary excretion represents the most important route of chemical, drug and waste elimination in the body. Normal glomerular filtration rate is determined by the endothelial fenestrations or pores formed by the visceral layers of the specialized endothelial cells of the glomeruli. The filtrate is then collected in Bowman's capsule. Reabsorption follows in the proximal convoluted tubules, that is PCT, loop of Henle and distal convoluted tubules, DCT. The collecting ducts have limited reabsorption and secretion capabilities. Control of the transfer of molecules back to the capillary circulation or elimination in the final filtrate depends predominantly on reabsorption and secretion processes. As with absorption through biological membranes and distribution in physiological compartments, urinary elimination of toxicants is guided by ionic and non-ionic principles. The mechanism of this therapeutic intervention relies, in theory, on the alkalization of the urine, rendering the acidic drug ionic, water-soluble and more amenable to elimination. In practice, however, the buffering capacity of the plasma coupled with a large dose of the sedative or hypnotic makes this a treatment modality of last resort. Next is fecal elimination. Fecal excretion, the second major pathway for the elimination of xenobiotics, is a complex process that is not as well understood as urinary excretion. Excretion of toxicants by way of the feces can result from direct elimination of non-absorbed compounds in the gastrointestinal tract, from delivery to the gastrointestinal tract through the bile, and from secretion into intestinal luminal contents from the enterocytes. In addition to undigested material, varying proportions of nutrients and xenobiotics that are present in food or are ingested voluntarily, that is drugs, pass through the alimentary canal unabsorbed, contributing to fecal excretion. The physiochemical properties of xenobiotics and the biological characteristics that facilitate absorption were discussed earlier in this module. Although most chemicals are lipophilic to some extent and thereby available for absorption, it is rare for whole of a compound to be absorbed. However, some macromolecules and some high molecular weight compounds that are essentially completely ionized are not absorbed at all. For example, the absorption of polymers or quaternary ammonium bases is quite limited in the intestine. Consequently, most of a dose of orally administered sucrose polyester, cholestyramine or paraquat can be found in feces. The non-absorbed portion of xenobiotics contributes to the fecal excretion of most chemicals to some extent. One other factor contributing to fecal excretion is intestinal secretion, which likely occurs by passive diffusion out of enterocytes or via exfoliation of intestinal cells during the normal turnover of this epithelium. Next is biliary elimination. The biliary route of elimination is a significant source contributing to the fecal excretion of xenobiotics and is even more important for the excretion of metabolites. The liver plays an important role in removing toxic chemicals from blood after absorption from the gastrointestinal tract, because blood from the gastrointestinal tract passes through the portal circulation through the liver before reaching the general circulation. A compound can be extracted by the liver, thereby preventing its distribution to other parts of the body. The liver is also the main site for biotransformation of toxicants, and metabolites may be excreted directly into the bile. In this manner, the liver can remove xenobiotics and their metabolites before entering the general circulation. Furthermore, xenobiotics and their metabolites excreted into bile enter the intestine and may be excreted with feces. However, if the physiochemical properties favor reabsorption, an enterohepatic circulation may ensure. Toxic chemicals bound to plasma proteins are fully available for active biliary excretion. The reasons that determine whether a chemical will be excreted into bile or into urine are not fully understood. Glutathione and glucuronide conjugates have a high predilection for excretion into bile, but there are marked species differences in the biliary excretion of foreign compounds with consequences for the biological half-life of a compound and its toxicity. It is therefore difficult to categorize species into good or poor biliary excretors, but in general, rats and mice tend to be the better biliary excretors than are other species. Furthermore, organic anions and cations are biotransformed and actively transported into bile by carrier mechanisms similar to those in the renal tubules. Chemicals ingested via oral exposure enter the hepatic portal system and liver sinusoids, only to be extensively cleared by the liver. This toxicokinetic phenomena is known as hepatic first pass elimination. According to the principles of elimination, compounds may be classified based on their bile to plasma ratio, outlines the classification of representative chemicals based on this ratio. Substances with high bile to plasma ratios are readily eliminated in bile. Next is pulmonary elimination. Substances that exist predominantly in the gas phase at body temperature are eliminated mainly by the lungs. Because volatile liquids are in equilibrium with the gas phase in the alveoli, they may also be excreted via the lungs. The amount of a liquid eliminated via the lungs is proportional to its vapor pressure. A practical application of this principle is seen in the breath analyzer test for determining the amount of ethanol in the body. Highly volatile liquids such as diethyl ether and certain volatile anesthetics, example nitrous oxide, are excreted almost exclusively by the lungs. No specialized transport systems have been described for the excretion of the toxic substances by the lungs. Some xenobiotic transporters have been identified in the lung, but overall compounds excreted via exhalation in the lung are most likely to be eliminated by simple diffusion. Elimination of gases is roughly inversely proportional to the rate of their absorption. Therefore, gases with low solubility in blood such as ethylene are rapidly excreted, whereas chloroform, which has a much higher solubility in blood, is eliminated very slowly by the blood. Most gases aerosolized molecules and volatile liquids are eliminated by simple diffusion via exhalation. Next is mammary gland elimination. The mammary glands are modified pseudoriferous glands that produce a lipid-laden secretion, that is breast milk, that is excreted during lactation. The lobules of the glands consist of alveoli surrounded by 1 or 2 layers of epithelial and myoepithelial cells anchored to the well-developed basement membranes. Unlike the blood-brain barrier and placenta, these cells do not interpose a significant barrier to transport of molecules. In addition, breast milk is a watery secretion with a significant lipid component. Consequently, both water and lipid-soluble substances are transferred through the breast milk. In particular, basic chemicals readily accumulate in the weakly acidic or alkaline environment of breast milk. Accordingly, nursing infants are exposed to almost any substance present in the maternal circulation. Other eliminations. Elimination of chemicals through saliva, tears, hair follicles and sudoriferous, watery or sebaceous oily sweat glands is a function of plasma concentration. Elimination is also influenced by liver metabolism, passage through the intestinal tract and pulmonary system, and by limited access to local glandular secretions. Thus, secretion of compounds through mucous, ophthalmic and dermal glands is not significant. In forensic toxicology, however, certain chemicals may be detected in minute concentrations in hair follicles and nails when plasma levels seem to be non-existent. Chronic arsenic and lead poisoning have been detected by measurement of the metals bound to hair follicles and hair shafts. We will conclude this module with summary. The ability to efficiently eliminate toxic materials is critical to the survival of a species. The complexity of toxicant elimination processes has increased commensurate with the increased complexity associated with animal form. For unicellular organisms, passive diffusion can suffice for the elimination of toxic metabolic wastes produced by the organism. Similarly, as exogenous toxic materials derived from the environment diffuse into a unicellular organism, they can also readily diffuse out of the organism. For a chemical to be eliminated from the body at a site of elimination, that is kidney, that is distant from the site of storage, that is adipose tissue, or toxicity, that is brain, the chemical must be transported from the site of origin to the site of elimination. The kidneys are the sites of elimination of water-soluble chemicals that are removed from the blood by the process of reverse filtration. Liver cleanses the blood of various endogenous and foreign molecules. The liver biotransforms the endogenous as well as exogenous materials by reducing their bioreactivity and preparing them for elimination. It eliminates wastes and foreign chemicals through biliary excretion. The study of drug disposition in physiological compartments is traditionally referred to as pharmacokinetics.